Hello everyone, this is video number 4 for the urinary system, and the main topics that we'll be covering are shown on the outline here in red. So one topic is a description of the two main types of nephrons, cortical versus juxtamedullary. What are the differences? So we'll kind of go over that. And then we're going to start in talking about the physiology. How do nephrons work? And how does blood filtration, all those processes work. And for this video, I'm just going to give you the overview of that. And in the next video, we'll start going through the details of all of the processes involved later. Okay? So, this slide is describing the two main types of nephrons and that are seen in the kidney. So we're zoning in this part of the kidney. This is the cortex, the outer stripe, and here is the medulla. So we're looking at basically right at the border between the cortex and the medulla. And that's what we have here, the border between the cortex up here and the medulla down here. And one type of nephron is called a cortical nephron, and the other is called a juxtamedullary nephron. Okay, now, a few of the main differences between them I will summarize in this slide and the next couple slides. So one is where they are located in the kidney. The cortical nephrons are mostly in the cortex. So that includes the glomerulus, glomerular capsule, the convoluted tubes. The loop of Henle does dip down somewhat into the medulla, however. Okay? So cortical, mostly in the cortex. The juxtamedullary nephron is positioned much closer toward the medulla, or the border with the medulla anyway. In fact, the term juxtamedullary means next to the medulla. So the glomerulus, glomerular capsule, proximal and distal convoluted tubules are still in the cortex, but now in a juxtamedullary nephron, the loop of Henle dips way down deep into the medulla. Okay, so that's one difference. Now, you may have noticed in that previous slide also that there are two different capillary beds that were shown. Let me just go back to that slide. Right. So one capillary bed is the glomerulus, and then there's another capillary bed shown here. And they are separated only by the efferent arteriole. Here's afferent bringing blood into the glomerular capillaries, efferent bringing it out. And then there's a second capillary bed, and then blood is drained through that, from that, and is going to eventually exit from the kidney. And so that's the situation in the cortical nephron. This second capillary bed is called the peritubular capillaries. And the juxtamedullary nephron, you have the same structure, afferent arteriole, glomerular capillaries, efferent arteriole. However, the next capillary bed is mostly surrounding the loop of Henle. So this capillary bed has a different name. It's called the Vasa Recta. Let's take a look at the next slide here, which shows the basic description. Two capillary beds separated by the efferent arteriole. The first one is in the glomerulus, and that's where filtration occurs. The second one is either the peritubular capillaries or the Vasa Recta, depending on what type of nephron is involved. All right, so this is showing for the cortical nephron, it's glomerular capillaries, efferent arteriole, and the peritubular capillaries. These peritubular capillaries are going to be involved in taking up materials exiting from the cortical, from the renal tubule. Okay. For the juxtamedullary nephron, the efferent arteriole is going to lead into this Vasa Recta. So the Vasa Recta is also going to be involved in taking up stuff from leaving from the tubule. However, it's mostly surrounding the loop of Henle, so it's involved with a different part of the nephron. Okay. So that's the second difference. One is position of the nephron. Second is, what is the second capillary bed involved with the nephron? Peritubular for cortical nephrons, Vasa Recta for the juxtamedullary nephron. Right, just a summary of the peritubular capillaries and where they're coming from. So they're rising from the efferent arteriole, and they're closely surrounding especially the proximal and distal convoluted tubules. These capillaries are extremely porous, and they're going to be taking up materials that are exiting from the renal tubule. Peritubular capillaries are found in the cortical nephrons surrounding the PCT and the DCT. Okay, here it is. The Vasa Recta, on the other hand, these are capillaries arising from the efferent arteriole and juxtamedullary nephrons, and they surround the loop of Henle, and they're going to take up material exiting from the kidney tubule at the loop of Henle. All right, so this table is summarizing the main differences between the cortical and the juxtamedullary nephron. I didn't mention this one. Cortical nephrons are much more abundant, making up about 85% of the total of all nephrons, whereas juxtamedullary, the remaining 15%. That should read 15%. Cortical nephrons mostly in the cortex, whereas juxtamedullary nephrons are closer to the medulla. The second capillary bed in cortical nephrons is the peritubular capillaries surrounding the PCT and the DCT. In the juxtamedullary nephron, it's the Vasa Recta surrounding the loop of Henle. And then the main difference in function between these, well, the cortical nephrons being most abundant are carrying out the bulk of blood filtration. So they're kind of the workhorses of the kidney. The juxtamedullary nephrons have a more kind of specialized function. They come into play especially when you're dehydrated, and they're involved in making more concentrated urine, meaning urine with less water content. And so they're saving that water to maintain blood volume if you're dehydrated. All right, let's go on to the second main topic of this video now, and we're switching into a discussion about physiology. How do nephrons work? So you have all these nephrons, more than 1 million in each kidney, and they're adjusting the concentration of your blood and the volume of your blood. And how do they do that exactly? So what is the process that's involved? And how does urine form during this process? So there are 3 main steps involved, shown on the slide. One is glomerular filtration. This is happening at the glomerular capillary bed. Another is tubular reabsorption. Tubular reabsorption happens in the kidney tubule. And another is tubular secretion, also happening at the kidney tubule. I made a few short videos to describe each of those, so let's take a look at these, starting with glomerular filtration. And these are happening at different places along the nephron. The first one is called glomerular filtration. And glomerular filtration is movement of fluid out of the glomerular capillaries into the capsular space. Okay, so out of the blood into this space just outside of the capillaries that's in the wall, inside the wall of the glomerular capsule. Okay, that's the first thing. Glomerular filtration is an extremely high volume process. You filter, that is, you move fluid into the capsular space 60 times your total blood volume every day. So that means about 2.5 times your total blood volume every hour. Extremely high volume process. The amount of fluid that's moving out of your bloodstream into these renal tubules. Okay? This process is driven by a pressure gradient. We'll talk more about that later, exactly how that works. But there's a pressure difference between the inside of this capillary and the capsular space, and that's what's pushing fluid out. Okay? The fluid that exits into the capsular space. So this is not everything that's in blood. It's basically water and small solutes. Large things like formed elements, red blood cells, white blood cells, platelets do not get out. They stay in the blood. but blood plasma, which is mostly water anyway, and small solutes like ions, glucose, amino acids, and small waste molecules, they do get out. So they pass into what's called the filtrate. If it moves into this capsular space, it's called the filtrate. Okay, and so that's the first process that's involved. Extremely high volume process, a large amount of fluid and dissolved solute moves out of the bloodstream. And what's the fate of that? Well, it's going to travel down the kidney tubule, the nephron, and whatever gets all the way through the tubule is going to end up in the urine. So you could probably guess, if you're filtering such a high volume of material out of the blood, not a lot of it is going to end up in the urine. So if our total blood volume is 5 liters, and we're filtering 2.5 times that, so that's 12.5 liters per hour, and we only produce maybe 100, 200 mils of urine. All right, let's go on to the second process, tubular reabsorption. Glomerular filtration, materials are moving out of the bloodstream into the kidney tubule and the capsular space. What happens in tubular reabsorption? The second main process that happens is called tubular reabsorption. After filtration occurs, fluid moves from the capillaries into the capsular space, and then there's a passageway out of the capsule into the beginning of the kidney tubule. The beginning part, this proximal convoluted tubule, the nephron loop or loop of Henle, and the distal convoluted tubule, which eventually connects to the collecting duct. Okay, as the fluid moves through this tubule, and by the way, the fluid is named tubular fluid inside the tubule. The filtrate here now is the tubular fluid. As it moves through, much of what passed out of the bloodstream is going to be reclaimed back into the bloodstream. So material is going to move from inside the tubule back into the bloodstream. And there's a set of capillaries that surround the proximal and distal convoluted tubules. I didn't show it for the distal, but they're called the peritubular capillaries. These are present in cortical nephrons. Okay? So the process of tubular reabsorption is moving of materials from inside the tubule back into the bloodstream. Filtration is moving it out of the bloodstream. Tubular reabsorption, moving it back into the bloodstream. So this process, like filtration, is an extremely high volume process. You filter in both kidneys about 180 liters per day. You form that much filtrate, 180 liters per day. You produce only about 1 liter or so of urine. So everything but that 1 liter must be reabsorbed. You reabsorb 179 of the 180 liters that filtered. So also an extremely high volume process. Okay? It's going to happen along the proximal convoluted tubule, a little bit in the loop of Henle, distal convoluted tubule, but the main site of it in terms of volume is in the proximal convoluted tubule. Now this process is going to require active transport of molecules out of the tubular fluid to get across the cells that line the tubule wall. Okay? So you have to move these molecules across 2 plasma membranes, the membrane on the luminal side and the membrane on the outside of the tubule. And then once it's out, it can enter into the interstitial space and enter into these capillaries. Most of the filtrate is reabsorbed, so there's a lot of water that gets reabsorbed. Some water is going to end up in the urine, but useful molecules like glucose and amino acids, they're small. They can filter out, but they're going to be completely reabsorbed, and normally you have none of those in your urine. All right, so that's the second main process. First one, filtration, movement out of the bloodstream into capsular space and into the tubule. Second one, tubular reabsorption, moving out of the tubule back into the bloodstream, reclaiming it. Okay? All right, so that was the second main process, tubular reabsorption. Let's look at the third and final process called tubular secretion. All right, the third process that happens in urine formation is called tubular secretion. Tubular secretion moves materials from the capillaries, the peritubular capillaries, into the tubule. So materials that have, let's say materials didn't get filtered here, they pass through the efferent arteriole into the efferent arteriole, and then the next stop is through the peritubular capillaries. This is a second chance for some material that didn't get into the tubule originally by filtration to get in through this process, tubular secretion. Tubular secretion, unlike the other two processes, is a relatively low volume process. This happens to kind of adjust, fine adjustment of blood volume and solute concentration, and also acid-base adjustment to the blood. So that process is mostly happening in the distal convoluted tubule and also somewhat in the collecting duct. And that process can be regulated by hormones. And that's the main site of regulation, is the distal convoluted tubule and the collecting duct. We'll talk about hormone regulation a little bit more later on. All right, so that's an overview of the three main processes involved in urine formation by the kidney nephrons. So this slide is just basically reinforcing that information. Three processes here are color-coded. Glomerular filtration is exit of fluid from the glomerular capillaries into this capsular space. Okay? The capsular space is continuous with the renal tubule. So that leads to the second main process, tubular reabsorption, and that is movement of materials out of the tubule, claiming useful stuff, glucose, amino acids, a lot of water, some ions, things like that, back into the bloodstream. Okay? We don't want to lose everything that we filtered, so we're going to reclaim most of it. Tubular secretion, shown in yellow here, is another chance for stuff that didn't pass into the tubule by filtration to get into the tubule by another way. So it can move from the capillary bed, peritubular capillaries, into the convoluted tubules, or can also move from the Vasa Recta, if this is a juxtamedullary nephron, into the loop of Henle. And that's a relatively low volume process for fine adjustment of blood volume and concentration, pH, things like that. Whereas these two are high volume processes. All right, just a note about the names of the fluids. It depends on where the fluid is. If the fluid has exited from the bloodstream in the glomerular capsule and is sitting in the capsular space, it's called the filtrate. If the fluid is passing through the renal tubule, it's called the tubular fluid. Once the fluid passes through the collecting ducts and gets to the renal papilla, then it's going to enter the minor calyces of the renal pelvis. So at that point, it's called urine. And that's all shown also kind of diagrammatically on this slide. Filtrate, that's what's happening here in the renal corpuscle, the capsular space surrounding the glomerular capillaries. Tubular fluid, any fluid traveling through any of these tubules, and even the collecting duct. But here, once it transitions from the end of the collecting duct at the renal papilla, this is the minor calyx in the renal pelvis. So this is the calyces, and that's where urine is collecting, and eventually is going to empty into the ureter.